Okay, so we just went over audition. Now let's talk about equilibrium or our sense of balance. Equilibrium is mediated by the vestibular apparatus. The vestibular apparatus consists of two main structures. The first is these right here, the semicircular canals. Okay, the semicircular canals are responsible for detecting head rotation. And then we have these structures right here, the otolith organs, the utricle and saccule. Don't worry about those names. But those organs are responsible for detecting linear head movement and head position. And I just want to point out, again, they're in the inner ear and they're connected to the cochlea, which we just talked about. And all of these structures are fluid filled. So let's take a look at how these structures work. First first the semicircular canals. So we have three semicircular canals and each one lies within one plane of movement, okay, or one axis of movement. So we have one semicircular canal that lies in this plane. And remember that it's fluid filled. So what happens is when we move our head back and forth like this, like we're nodding yes, then the fluid in that canal is going to kind of slosh back and forth, okay? We have another one that is in this plane. So when we turn left and right like this, the fluid in that canal is going to slosh back and forth. And then we have another one in this plane like this, so that when we tilt our head left and right, the fluid in that canal is going to slosh back and forth, okay? So that fluid movement is what we are able to detect. And the way that we detect that is that, actually going back, at the base of each of these semicircular canals is kind of a bunch of structures, but in the kind of the middle of that bunch of structures is this gelatinous structure called the cupula. And inside the cupula, we have these hair cells. What happens when the fluid in one of our semicircular canals moves back and forth is that causes the cupula to move back and forth, and that causes the hair cells to bend, the cilia on the hair cells to bend. And then just like with the hair cells that we saw in the cochlea for audition, the bending of these hair cell cilia is going to lead to opening and closing of mechanically gated ion channels. And as you know, when we open or close ion channels, that's going to change the ion movement across the membrane. That's going to change the membrane potential, and that is our signal that our cell can detect. Okay. All right, so that was the semicircular canals. Now let's take a look at the otolith organs. Okay, so first of all, I just want to remind you the otolith organs are right here, kind of at the base of the semicircular canals. This is what it looks like inside of an otolith organ. We have just sort of this tissue right here. Inside that tissue is are these hair cells that are embedded in there. On top of those hair cells, we have a gelatinous membrane called the otolith membrane. And then on top of that membrane, we have these little crystals called otoliths. So what happens when we move our head? Let's take a look at that. Okay, so first I just want to remind you that the otolith organs the otolith organs are responsible for detecting linear acceleration and for head tilt, okay? So what happens is when our head is in a neutral position, things just kind of look normal. The otoliths are just hanging out here on top of this membrane. When we tilt our head, what happens is gravity causes our otoliths to move. That moves the membrane, that bends the stereocilia on these hair cells. And then again, that causes mechanically gated ion channels to open. That's going to cause a graded potential after those ions move across the membrane, okay? So this diagram shows how head tilt is detected, but basically the same thing is going to happen if our head is like accelerating in space. The otoliths will move and cause those stereocilia to bend.